Okay, so now we know some of the anatomy of the skeletal muscle. We can start to think about the physiology of how muscle contraction works. So you might be able to watch, you're probably watching this video before class, or maybe not, but um, what we want to remember is that we're going to have a couple of things extra that we're going to have to draw and label before we're actually completely ready to do muscle contraction, okay? So first thing is I gave you this picture that I drew out because it's a lot of stuff to draw and I wanted to cut down the length of the video. So it's just easier to give you this picture. We're going to label a couple of things that you should already know, but if you don't know them because you didn't look at the other stuff about synaptic transmission, then maybe you're confused. So the thing we want to think about is this is the axon terminal and the synapse for what's called a somatic motor neuron. And if you're like really confused by that, remember how the nervous system, the peripheral nervous system, is going to have two divisions, a sensory division and a motor division. And then the sensory and motor division are further subdivided into somatic and visceral. What those terms help us realize is, excuse me, there's going to be neurons that specifically supply like somatic motor or visceral motor, or somatic sensory or visceral sensory. So if we're going to contract the skeletal muscle, remember that was part of our somatic motor division. So we're going to have a neuron that just sends the somatic motor message to a muscle, to a skeletal muscle in this case. So here we have our synapse. And then we're going to see we have our synaptic cleft. Okay, remember that's just the space that goes between the synapse and the receiving cell. And in this case, our receiving cell is our skeletal muscle. So we can label this red line. This red line is going to be my sarcolemma. And then we're going to have to draw a couple of extra things here. Ooh, wrong color. We're going to have to draw a couple extra things. So here's the synaptic vesicles. And now inside of our synaptic vesicles, you want to write ACH, because remember, we're going to have acetylcholine inside of our synaptic vesicles, okay? On the sarcolemma, you're still going to have those red channels. Those red channels were our sodium channels, okay? So we're going to set all this stuff up. We have to have a sodium voltage gated channel on our synapse, and we're going to have more sodium on the outside of our neuron. And we're also going to have sodium at a higher concentration outside of our muscle. So remember, red is for sodium and green is for calcium, just like that. We should see that, and then I'll make this orange acetylcholine to help us out a little bit, okay? We want to remember that our muscle cell is negatively charged on the inside, all along like that, and it's positively charged on the outside. It's positively charged on the outside because remember, we have all this sodium just sitting out here, okay? Potassium will be the same as being higher on the inside than it is on the outside, but we're gonna negate potassium. We're just gonna make this a much more simplified process. It's not simplified, but this will simplify it just a little bit for us. So we got ACH. Just made my synaptic vesicles a little bit small, okay? So that's our setup. So right now, our muscle cell, our sarcolemma is negative 70 millivolts on the inside. We have lots of sodium on the outside, lots of calcium on the outside. Your brain is going to send, your brain or your spinal cord are going to send a message down your somatic motor neuron. And when it does, remember, an action potential is created that action potential goes down the axon terminal, and as that positively charged message reaches our calcium voltage gated channel, our calcium voltage gated channels are gonna open. When our calcium voltage gated channels open, sorry, I can't see my white in order to open my channels. Okay, you get the picture. When my calcium voltage gated channels open, because calcium is higher outside than it is inside, calcium starts to move inside of the synapse. So this is all stuff we learned before. The calcium binds to the synaptic vesicles, and when it does, it causes the synaptic vesicles to fuse with the synapse. And when it fuses with the synapse, what that means is that little pouch or that little pocket is going to open up. And when it opens up, it's going to release acetylcholine. So now you have sodium and acetylcholine that's going to be high outside of the cell, okay? The acetylcholine is going to come and bind to its receptor on these sodium channels that are in the sarcolemma. And when that happens, those sodium channels are going to pop open. I mean, they don't pop open, but they open, okay? They open. So we're going to think about that's going to open. That's going to open. That's going to open. Just redraw my line a little bit like that, okay? Now we already said sodium is high on the outside. So since sodium is high on the outside, this is going to cause sodium to start to enter the cell. And now the cell is going to become positively charged. In this case, there is no threshold. This is all or nothing. You open your sodium channel, you're contracting your muscle. There's no like, oh, you didn't reach enough sodium, so you're not going to open any more channels. No, you open one channel and you're going to start the whole process, okay? Since sodium started to come in, it's going to cause sodium voltage gated channels along the sarcolemma to also open. So basically, more and more sodium enters. And that means our sarcolemma becomes more positively charged, okay? So here's where it's going to be different than what you learned last time. This is where we stopped. We created an action potential. Basically, we create an action potential along our cell membrane. But now, excuse me, because we have a muscle fiber, what's going to happen is once we reach the T-tubule right here, that positive charge goes down the T-tubule. So this all becomes positively charged as we go deep inside of the cell. That is going to cause the calcium that is in the sarcoplasmic reticulum. That's what this blue thing is. I should have labeled that. Let me do that really quick. I didn't really set up all my stuff, I guess. So this is my sarcoplasmic reticulum. Okay. So that positively charged message, or the positive charges, are still entering the cell along the T-tubule. And once that occurs, this is going to cause calcium to be released. So calcium gets released out of the sarcoplasmic reticulum. Okay. The next thing that's going to happen is what calcium is going to do to our sarcomere. So if you hadn't figured this out, this is me drawing my sarcomere here. So the calcium being released is going to bind to troponin. Troponin then causes, that binding of calcium to troponin causes tropomyosin to twist. And when it does that, it's going to open the myosin binding sites on actin. When that occurs, the myosin is going to bind to actin and pull actin, I should say pull actin towards the M line. So the Z disc will move towards the M line and our sarcomere is going to get smaller. Okay. Now, for muscles to relax, what's going to happen is the acetylcholine has to be broken down here at the receptors. Once the acetylcholine gets broken down, the sodium channels close, calcium gets sucked back up into sarcoplasmic reticulum, your sarcomere bounces back to its normal size, and your muscle goes back to relaxation state instead of being contracted. Okay. How does acetylcholine get broken down? There's an enzyme that breaks that down in case you're interested in knowing about that. So the next part I'm going to do is I'm going to pick up from when calcium got released and show you what happens to the sarcomere, what's going to happen at the sarcomere in order for the muscle to contract.